Recording is on.
This is the first of a series of lectures devoted to looking at some biology problem from the perspective of physics. In this case, we are going to look at mutations. Data on mutations come mainly from two sources. The first is the long-time evolution experiment with Escherichia coli by Professor Lenski, and the second is let's say an experiment also, or a recopilation of data from 100,000 people, Europeans, and they collect copy number variation in 3 generations. There is grandfathers, fathers, and sons. The data suggests that in the model we shall include at least 2 independent processes, single point mutations and large chromosomal rearrangements. And let's say abstract view of these processes can be described with the Markov chain, in which the configuration of the chain at time 3+1 is expressed in terms of the configuration at time t plus a modification, that is the mutation itself. This modification contains 2 components mainly, single point and large rearrangements, and we shall indicate the time rates and the length distribution function of this later component, that is the distribution function as the distribution as a function of the length of the mutated fragments. Notice that we introduce a unique term to describe large rearrangements, in spite of the fact that there are many processes, non-processes, in this or related to this large rearrangements. Notice also that we cannot describe in a unified way the single point mutation and rearrangements, and this, from our point of view, is suggested by the observation in the experiment of a mutator phenotype, which increases by 100 times the rate of single point mutation, but does not affect large rearrangements. A complete description of mutations shall include, besides the rates and the length distribution function, the actual site and the modification of the DNA change. Our description is not complete, that is, we give a partial description of mutations. In addition, the data itself is incomplete. Usually, there is a good description of single point mutation and a good description of very large rearrangements. The intermediate and low length range is poorly described. Our model is a kind of basal one. There is no effects coming from selection of 3 forces are included in the model. The data come from all these papers. We give the precise sites here, and partially this data is posted in this repository we created, and also the Python routines to process the data are available there. Notice that, to the best of our knowledge, there is no results concerning the length distribution function of mutations. This is the main results of the paper, indeed. Here, this first figure we compare, or we put in the same, in a single figure, single point and large rearrangements, single point mutation and large rearrangements. The purpose is to show that a mutator phenotype affects by 100 times the rate of single point events, which cannot be represented here because it's out of the range of the figure. That is, the mutator phenotype appears at roughly at generation 27,000, but the large rearrangements are not affected by this mutator phenotype. It means that these two processes are radically different. There are also here, and we wrote some details, but details can be found in the paper also. Okay. The next figure corresponds to the length distribution function. This first upper panel corresponds to a particular population of the 12 population involved in or followed in the long time evolution experiment with the Escherichia coli. And these results indicate that the tail of the distribution function is a power or shows a power-like dependence. The exponent is 0.42 in this case. The number of events is very small. It's not a data with a good statistics, but nevertheless, it shows a very good. It can be fitted with a power-like dependence. This statistics improves when we put together the 12 population. There is data from the 12 population followed in the long time evolution experiment. In this case, the exponent becomes a number near 1/2. Notice that there is a saturation in this low length region. This saturation may have different causes. It could be simply that the distribution function saturates indeed, or it could be related to the fact that the experiment cannot reliably detect mutations in this range. Okay, the second saturation, observed saturation is here at probably at around 2 megabase pairs. That this is related to the length of the circular DNA for Escherichia coli, which is near 5 megabase pairs. Okay, here the lower bound is around 5 kilo base pairs. This top panel shows that we can fit the data in the entire range by using a Levy function. This Levy function has the power-like dependence at large L and saturates at low lengths. In addition, this Levy function has the property of the Gaussian in the sense that they respect the central limit theorem, that is, the sum of independent events is again a Levy process. And that's why we choose this function. Okay? All these comments I already made. This is the comment about stability. And here I should also stress this point that the data we have is incomplete. It's not there is maybe additional data that could be found in the future could suggest including other processes with the dependent time rates. For example, small length in delta. That's a possibility or actual possibility. Okay. This next figure is related to mutations in human germline cells. Here is a description of this kind of mutation. This left panel corresponds to number of mutation, which is more or less the rates. They are rates, that is. And this right panel corresponds to the characteristic length, that is. As the length, the characteristic length increases, the rates of this mutation decreases. That is, they are inversely proportional. This next figure is a similar figure to the previous one for Escherichia coli. That is, we show for copy number variation. This data is for copy number variation. I mentioned that there are 3 generations: grandfather, fathers, and sons. And the copy number, that is, we know the author of this paper report de novo variations, or de novo mutation, that is, mutation found in the son or found in the grandson. And for this, there is a range in which there is a power law behavior of the distribution function. The power is nearly 3/2. And again, the data can be fitted in the whole range with the Levy function, Levy 3/2 function. Let's mention that these copy number variations are low frequency mutations. That is, in the population they show low frequencies. Then they are mainly deleterious or neutral in character. And the authors show that there is a strong correlation to diseases. In the data range, they show a scaling, as I mentioned. Okay, the bottom panel is supposed to show that there are fluctuations in the low length scales. Nothing more. Okay. Then let's conclude. First, the possibility of describing large rearrangements by means of a unique process suggests that there is a general law in the basis of such processes. We shall stress also that there is an independent work by Polychronopoulos and co-authors where they report a length distribution function of spacers, that is the distances between highly conserved fragments in the DNA. Possibly this distribution function or follow from our model of mutation also. From a general perspective, the combination of single point mutation and a power law distribution of large rearrangements is optimal for solving the DNA configuration space in the seek for maximal fitness. This is an abstract view, but it could be quite reasonable. In fact, there are algorithms, there are programs that use this strategy for finding minima of potentials. It is known that the best exponent we can use is a Levy function with exponent 1. We obtained 1/2 for bacteria and 3/2 for humans. It's interesting that the optimal is in between these two numbers. The results are preliminary because the data is scarce, and as mentioned, this is incomplete. There is an interesting question here relating to the following: whether this, let's say, change from 1/2 to 3/2 is a consequence of evolution in some way. This also is, of course, a very interesting question. Also, the possibility of describing somatic mutations, which are very important in cancer, for example. I believe that the next step of our work is to take data for somatic mutation, try to fit them to check that they follow also power laws as in the one we checked already. Okay, thank you very much.